All right, so I'm Autumn. I'm a senior bio major, chem minor here at Hartwick. I have been working with Dr. Carr for the past 4 years on trying to understand motility in the subsurface ocean. Before I get started, I wanted to say thank you to Dr. Allen and Judy Friedman, and thank you to Hartwick, and thank you to NSF for being able to provide funding for this research project. So a brief overview, I'm going to talk about some background information and introduction to my sample site and an introduction to the subsurface ocean. Then I'll talk about some previous research that I've done over the past 4 years here at Hartwick. And then I will talk about my big plan that I have had for this thesis. And then I'll talk about the progress with that plan and some troubleshooting. So here's my introduction to the Juan de Fuca Ridge. So this is an area in the Pacific Ocean. It's right off the coast of Oregon. It's this yellow box right here. That is where we got our samples from. And just know I'll be abbreviating the Juan de Fuca Ridge, the JDFR. So then I'm going to get into the actual marine subsurface. So this area is so water comes down from the bottom of the ocean, and that water is filtered through these outcrops in the ocean floor. These are called hydrothermal outcrops or hydrothermal vents. So that cold water from the ocean is coming through and filtering through this area in the ocean floor called a crustal aquifer. So that water, that cold water from the ocean, is then heated in this area by the lithosphere. And then that hot water is then transferred back out into the ocean. So this area, this crustal biosphere, is extremely, extremely hot. There's not a lot of nutrients. As we can see here, the dissolved carbon is very little. There's only 14 micromoles per liter. It's around 64 degrees Celsius, which is pretty hot. And this area doesn't have a lot of nutrients. This is its electron acceptor. So an important thing to know about the crustal biosphere is that there are 1 × 10⁴ cells per mil, which doesn't seem like a lot, but when you add all of the water in these aquifers, that comes to a lot of cells. So this is where we were sampling, where we got our samples. So like I said, this area in the subsurface has very little energy, but as we can see from this graph, this has taken samples from marine sediment, which is what this area is, and hydrothermal vents, which are those outcrops, and then the crustal aquifer, and that is where I have been kind of focusing my research. So sampling was done with these they're called corks, but they're basically these boreholes that are kind of like wells. They drill into the seafloor and can collect sediment, like is used here, fluids, which is what I have been using in my project, and they take a bunch of other data that we can try to use to understand what life is like in the subsurface. So my main focus has been on motility in the subsurface, primarily the crustal aquifer fluids. But this graph, Dr. Carr did some research previously, and it shows that it shows the abundance of motility genes in these marine metagenomes from the Juan de Fuca Ridge sample site. And as we can see here, the crustal aquifer has like it has about 2% of 2% abundance of motility genes in its metagenomes, and that may not seem like a lot, but comparing it to Salmonella or E. coli, they have about 2.5 to 5% abundance. So even though these microbes live in a very nutrient limited environment, they obviously have the genes for motility, and my project has been trying to understand how those are being used, and if they're actually being used, that kind of stuff. So some previous research I did starting my the summer after my freshman year, and then into sophomore and junior and senior year. So my freshman year, I looked at 42 genomes from the Juan de Fuca Ridge to search for any type of motility genes that they might have. So these circles represent the genomes that I looked at, so 42 genomes. And out of the 42 genomes, I found that 41 of them had some type of motility genes present. The only one I didn't find any present in was this methanopyri. But as we can see with these circles, the more filled in they are shows how complete the genome is. So say like 39, that would be very complete, but this one is not very complete, so that might explain why I didn't find motility genes in there. So from this, we know that these genomes have motility, some type of motility genes present. And it's important to note that these are the genomes that I looked at were for archaeal species, and that's what I focused my next research on. So then, sophomore year, I went to Bigelow Laboratories up in Maine and worked on a bioinformatic project trying to understand what types of motility genes these archaeal species from the Juan de Fuca Ridge had. So this is a phylogenetic tree that I made that summer. These orange dots are my Juan de Fuca Ridge species. I made this tree by incorporating some base tree from a paper I read that was talking about different appendage structures that microbes use for motility or biofilm production. So I added my sequences into their data, and this is the tree that I got. These red dots, these are a Hydrothermarchaeota species that Dr. Carr was interested in. I'm not going to really talk that much about those, but it's important to note. So each of these clades represent a different ATPase structure. So ATPase, those are used to build the actual motility structure. Those are like the building blocks of different motility structures. So it could be pili, flagella, or an archaeolum, which is a flagella for archaea. And this is what clade 3 represents. So some important takeaways from this tree, based on the Hydrothermarchaeota, where they're placed in the tree, they're placed pretty far back. So that could possibly show that these appendage structures were an early evolved trait. Then because my these Juan de Fuca Ridge orange dot sequences are in each of the 4 clades, it shows that many of these groups have more than one type of pili or motility structure. And it shows that these archaeal species have these external pili structures that they're using for motility or biofilm production. So then my big plan based on this information, I hypothesized that swimming and attachment via motility structures or appendages are important for life in the crustal subsurface. So then my big question was, what are these microbes attaching to or moving to? So my goal was to grow the only model organism from the Juan de Fuca Ridge. It's called Archiglobus sulfatic calidus. And then once I got that growing, I wanted to induce appendage expression, either primarily through biofilm production. So my first big project with this was based on a paper I read that used metals to induce biofilm production in a similar species called Archiglobus fulgidus. So I added to liquid seawater media ammonium chloride and copper chloride to do different batches of media, and I filtered them and looked at them on slides. So as we can see, with the ammonium chloride, it didn't seem like there was any biofilm production, but with the copper chloride, these kind of stringy parts, this cloudy stuff, that could be an indicator of biofilm production, because the stain that I use, Cyber Green, stains DNA. So when biofilms are being produced, they make an exopolysaccharide layer outside, and that does contain some DNA. So these kind of stringy areas could be that exopolysaccharide, those formations containing DNA. So based on that, I found another paper that focused more on trying to induce motility or biofilm production on an actual surface, not just in liquid media, because you can't really tell if these microbes are making a biofilm and attaching to something if it's just in fluid media. So this paper suggested that you use glass slides or glass cover slips to allow to give these microbes something to attach to. So I first tested these methods out with E. coli, and the E. coli worked really well. As you can see, all of this, this is on the slide, this is on the cover slip, that worked really well. And then when I went to work with the asulfatic calidus, it did not attach at all. I couldn't see anything on the cover slips or slides. So then I wanted to see if these microbes could grow on a kind of agar or gel. So my goal, like the experimental setup, would be to have slides in a petri dish with this agar. I used a gel and gum mixture. It's a powder that you can mix with water and it will solidify. I wanted to pour that onto the slides and then grow microbes, have them get growing separately, and then inoculate these plates with liquid media with the growing microbes. So that was my goal. Over the summer at Bigelow, we tried making these gel and gum gels in a vial, but it just didn't work well. It wouldn't solidify because these microbes have to be at high temperatures. It just wasn't solidifying well. But then I went back to the original plan of doing it in a petri dish, and this worked a lot better with allowing the gels to solidify. So you can kind of see this pink tint here. That is the gel and gum on the slides, and these are the slides that would allow the microbes to grow. in that saltwater, inoculated saltwater medium. So then the progress with that plan and then the troubleshooting I had to deal with. So I had major contamination issues starting in January. I haven't had contamination issues at all, but once I went to inoculate those plates from the previous slide, there was just so much contamination. So then I've been working since to kind of figure out where that contamination is coming from. I made different kinds of media. I tried growing different microbes. I tried making new media for that specific microbe, new media for my microbe. I did all kinds of things with media. I remade all of my staining solutions. So there's multiple steps in the staining process. So there's making, I made new solutions of the actual stain, new solutions to help that stain not fade, new solutions to make those solutions. And I use a lot of water to filter these slides so I can view them under the microscope. I autoclaved that water. I filtered it with I think it was like a 0.45 micron filter, and then I brought it down to a 0.2 micron filter, and the water was the issue. So there was contamination in the water, which was getting into everything else. So that was what was causing those issues. And I only found this out like a couple weeks ago. So it's been a long process trying to figure out what is going on. So from there, I want to say thank you. I am still working on getting these microbes to grow. I'm trying to get some contaminant-free cultures growing, and hopefully within the last week and a half of school here, I can get a contaminant-free culture. So thank you.